Center for Integrated Environmental Health Sciences and Department of Pharmacology and Toxicology welcomes you to our Environmental Health Science Series seminar. Our speaker for today is Dr. Michael Merchant, Professor of Medicine and Pharmacology and Toxicology at University of Louisville. Dr. Merchant conducts investigator-initiated NIH-funded research into development and progression of kidney disease, complications of end-stage kidney disease, as well as other diseases such as alcoholic hepatitis. Pertinent to his talk today titled Metal Toxicity in the Kidney, his lab is studying the role of arsenic and antimony on renal dysfunction and complications of end-stage kidney disease. Encompassed within these studies are multiple high-impact manuscripts published in top journals such as New England Journal of Medicine and the Journal of American Society of Nephrology. Dr. Merchant's professional career focused on protein chemistry, starting as a graduate student working with Dr. Collis Jaron at University of Arkansas, studying isolation and characterization of protein toxins from venoms of the black recluse spider and the timber rattlesnake. He transitioned from protein chemistry to proteomics research and evolved beyond to direct or co-direct multiple technology facilities, such as director of the Integrated Toxicomics and Environmental Measurement Facility Core for Centers for Integrated Environmental Health Science, and co-director of the University of Louisville Core and Clinical Proteomics Laboratories. Dr. Merchant's long collaborations and goals are to establish a modern translational omics research program with investigators in the US and also abroad. So please join me to welcome Dr. Merchant.
Thank you, Dr. Chen. And thank you, Dr. Kai, for the invitation. So I want to talk today about metal toxicity in the kidney. As you just heard, my primary appointment is in the Department of Medicine and in the Division of Nephrology. For about the past 20 years, I've kind of focused on kidney disease and focusing on human disease and then trying to translate it into an animal model or a cell culture model to maybe understand mechanisms. My interest in metals here at Louisville dates back to about 5-6 years ago with a consortium of individuals that I'll cite at the end of the talk that looked at lifetime exposure of mice in utero and through the F1 generation to arsenic as well as cadmium. My initial exposure to metals research was in last century in undergrad doing X-ray powder diffraction of coal fly ash and looking at the compositional analysis of that. And that was predating most computer algorithms that would search the spectra. So this was mainly going through large manuals of spectra, identifying components in the fly ash. What I want to do is kind of merge together my role within the Center for Integrated Environmental Health Sciences as a facility core director with my role as proteomics and kidney disease to kind of bring you into a work in progress. Let's see. Sure. Okay, so I believe I'm sharing now. Try not to share too much. So I don't have any disclosures. Absolutely. Thank you. So as part of an outline today, I will address the topic of metals in the kidney, look at some kidney disease variables associated with metals, address a current work in progress study that brings in kidney proteomics, metals, and sexual dimorphism in the response to metals. And lastly, future directions to mitigate some of the problems that we have with following the effects of arsenic on the kidney. So within metals in the kidney, I want to address renal function, markers of renal damage, and population data on metal nephrotoxicity. So for me, wanting to be a translational person, the first thing I would like to do is ascertain whether or not there's a significant impact of metals on renal function as it exists in the literature, and consider the variables that would influence the analysis of population style data. I think metals and their toxicity associated with carcinogenesis, teratogenesis, mutagenesis is very well established, and so there's a lot of compelling reasons to follow that. But with renal disease, it's less clear. It is evident, but it's less clear. So in the proteomic study that we're going to conduct, that we have conducted on arsenic as well as cadmium, the kidney is compartmentalized into a couple of different zonal regions. One zonal region is the cursor popping up? Okay, so we have the cortex of the kidney, which is kind of an outer zonal region where you have a lot of oxygen. And that's where most of the filtration of the blood takes place. That's where most of the microvascular beds are found that are responsible for filtration of the blood and generation of the urine. Then we have the medulla, which is a kind of a higher osmotic gradient zone, lower oxygen tension zone, so more low oxygen environment, almost hypoxic if you want to think of it that way. And then we have some other zones in here that are mainly for urine collection and diversion down to the bladder. So we tend to always drive our study on the cortex, mainly because most renal diseases, kidney diseases, are thought to originate at the level of the glomerulus, at that microvascular bed. Most, but not all. There are interstitial diseases that take place within the tubulo-interstitial compartment, those compartments that would be within these rays down through here. So whenever we conduct an animal tissue proteomic study or a transcriptomic study in the bulk sense, we tend to always isolate the cortex. And so, but to further elaborate on the structure, the functional unit of the kidney, I'd like to go through this figure, which is something that elaborates the nephron, the structure of the nephron. So we have a glomeruli, which is comprised of an afferent arteriole, a capillary bed, and an efferent arteriole, and that's how the blood is perfused into this microvascular bed. Then there's a capsule surrounding this area called Bowman's capsule that's lined by an epithelial cell type. The urine is filtered through the urinary pole into a tube lined with epithelial cells. So this is the tubular epithelium. And there are different functions of the epithelium along the nephron itself. And so these different epithelial cell types have roles in a kind of a zonal region that are associated with, for instance, protein reuptake whenever the filtration barrier of the glomerulus is damaged, or whenever there's a large degree of hypertension, a large amount of blood pressure coming in, when there's concussive damage, a variety of things, autoimmune diseases damaging the endothelial layer, the basement membrane, and the epithelial layer of this microvascular bed. You can have urine ultrafiltered across. That protein, you can have serum protein ultrafiltered across. That protein represents energy that your body expended creating or acquiring from the environment. So to conserve energy, there's a net need for the organism to reuptake that protein. So the first area of the proximal tubular compartment is lined with protein transporters, things that'll bind the protein and bring it back in so it can be degraded and put back into the bloodstream as amino acids or as whole proteins. Different sections along the nephron are concerned with regulation of that osmotic tension, so affecting salts, fine-tuning salts out of the blood into the urine, as well as water volume status. So in considering how a metal might impact a kidney, the metal as a small ion being freely ultrafiltered has the potential to impact the epithelium, the glomerulus, the vascular bed, and the epithelium along the entire length. So within the microvascular bed, that vascular pole right there is attached to a series of cells in the juxtamedullary macular densa region that are responsible for the productions of some types of hormones. And it's through the collective functioning of the myofibroblastic-like cells, the mesangial cells within this vascular bed, to respond to cytokines, cytokines, they will stretch or contract and regulate blood flow. So the normal renal function, the normal function of the kidney, is something that's going to impact a lot of other systems biologies. The role for the kidney is to remove waste, remove drugs, balance out the body fluids, release hormones, regulating blood pressure, modifying the active form of vitamins, vitamin-based hormones, and to control the production of red blood cells through the generation of erythropoietin. So I want to introduce this stage, even though it's fairly remedial for most of you, to something that I'm going to discuss later on when I mention how human data has been analyzed and some of the failures in the analysis. But to consider, whenever we discuss renal function, we may be looking at one feature of these affected by the metal, not all of these. So there tends to be a default in how we approach renal dysfunction, and there's some hidden problems with that. So kidney damage. I want to jump in and talk about kidney damage, because whenever you're addressing how a metal or how an environmental toxin may impact the kidney, there's a certain amount of default approach of people to examine a particular endpoint to measure the damage. And so this is a clip out of a movie called The Immortals, where you have the Titans battling the gods of Olympus, and kind of this never-ending battle. And within the field of nephrology, there's a similar battle that goes on about what the appropriate measure of renal damage is that we should be using. And so this is important to consider, because in that context, as time goes by and as those endpoints are tweaked and modified and improved, then the variables that we calculate, such as the estimated glomerular filtration rate, change, not because the serum creatinine changed, but the equations that transform it into a variable for renal function change. Then the whole landscape of how metals are correlated to disease change. So kidney damage is a set of staged levels of changes to the kidney that transform it from a normal function into a predisposition to disease, into a loss of renal function. So one of the things that we use as a standard for renal disease is how well the microvascular bed ultrafilters the blood. And by and large, people use a surrogate value of the serum creatinine as a measure of this value. The other way would be to actually inject somebody with a small molecule that's not going to be taken up by any tissue, it's not going to be metabolized, and it's going to be filtered straight through the glomerulus. And then at a specific time point, you sample the blood and the urine and measure that amount, and you'll get a fractional excretion rate. The other large way that we measure and evaluate kidney damage is to look at this histologically. So we sample the kidney, whether it's in a person taking a biopsy or it's an animal and collecting the kidney. We will section it and typically stain with a histologic stain that's going to show levels of fibrosis or sclerosis, or show amounts of inflammatory markers. So, and then the third way that's not illustrated here, and maybe you can infer it into this, which is accessing a biofluid, is to look at the levels of protein in the urine. And within the urinary protein measurement, you can speciate that out into blood, which is important for something like IgA nephropathy or damage, trauma, or you could speciate it into albumin, which is associated with most all renal diseases. So there's some confusion about serum creatinine. And serum creatinine is thought to be something that's produced in kind of a constitutive steady state process. And that process is uniform throughout your life. But we know that that's not true. We know that creatinine is largely produced, metabolized, and excreted by skeletal muscle. And so depending upon the amount of skeletal muscle you have, that's going to affect the steady state amount of serum creatinine in your blood. So this is just an illustration of a recent study where people used DEXA scanning and speciated people based upon cachexia, or loss of skeletal muscle, versus obese individuals, where you're not getting an accurate assessment of the amount of skeletal muscle that they had. So in this study, what they looked at was to take several hundred people, measure creatinine in their serum. They injected them with a dye molecule that's more associated with radiography. It's a technetium compound, radioactive, not radioactive, but it's, yeah, it's radioactive, sorry. And so they measured the fractional excretion of that and converted that into a measured GFR and compared it to serum creatinine levels. And what you can see is that there is a correlation between serum creatinine in the blood and the filtration rate, but it's actually not linear. It's only linear across a certain range of GFR, glomerular filtration rate. So if you're an adult in your 20s, you're pretty much thought to have a filtration rate. Your kidney filters how many liters of blood a day and produces about what volume of urine a day? Does anybody have a guess on that? 1.5 to 2 liters a day. Yes, exactly. So an average adult is going to produce about 1 to 2 liters of urine a day. So you're constantly ultrafiltering a couple hundred liters of blood throughout the day. You're producing a couple of liters of urine. What you want is to have this steady state disposition of analyte into the urine, have a steady state amount of the substance in the blood. You're going to measure that and try to correlate it to how well the kidney is functioning. So if you're an average adult, 20 to 30 to 40 years old, you're healthy, you're thought to have an ultrafiltration rate of 100 mils per minute. That's how well your kidney works. If it's higher than that, then that's called ultrafiltration, and that happens a lot in people with prediabetes. As you hit about the age of 40, you're going to start losing renal function, just as a normal course. You lose about 1% of renal function a year. That's a general thought. Anything more than that is considered to be kidney disease. So in this study, they then converted this measured GFR. They adjusted the GFR for the amount of skeletal muscle. And what they saw was that there was a tight correlation with the measured eGFR with the amount of skeletal muscle, and it really wasn't impacted by the amount of adiposity. Okay, so there's a direct correlation between your kidney function that's measured by a surrogate value and the amount of skeletal muscle. So in the past year and a half, 2 years, there's been this war within nephrology of what equations do we use to convert creatinine to a measured GFR. And the reason that this is appropriate is because that ability to estimate GFR impacts, for instance, when somebody's lost their renal function and they want to go onto a kidney transplant list, the amount of residual renal function can impact when they get placed on that list. So your ability to receive that interventional therapy is impacted by these equations that people use to calculate residual renal function. And that residual renal function, that metabolite, is based upon skeletal muscle. And there are some associations with high dense muscles with higher levels or lower levels of creatinine. So you can be an individual with a lot of skeletal muscle, poor renal function, but you can be estimated to have more or less renal function than you actually have at the level of the kidney. So this is the other data here is just some regression showing some dispersion in the data across certain ranges of GFR. The data gets tighter as you lose the renal function. That's just because the dynamic range of those measurements is compressed. This graph just simply is showing that the absolute error and the percent error is maintained using the skeletal muscle measurement across the different GFR ranges. So serum creatinine produced by the skeletal muscle, metabolized by the liver, excreted by the kidney, ultrafiltered by the glomerulus into the nephron. There for urine excretion, there are actually proteins, transporters lining the basolateral side of the tubular epithelium that the basolateral side of the tubular epithelium is in contact with the basement membrane. On the other side of the basement membrane is a perivascular bed. So there's an active transcellular transport of creatinine out of the blood into the urine. And so the functioning, the secretion of creatinine can be impacted by these transporters. So I'm wanting to introduce this to you at this level because some of the data that we observe in our proteomic data sets begins to speciate our samples based upon sex and sexual dimorphism. And one of the big features, or a feature that we've observed in our mouse kidney proteomes is differences, very dynamic differences in some of these transporters. And so how we measure renal function and we examine it between sexes, we can see differences possibly due to differences in a few proteins. So metrics and markers of kidney damage. The kidneys, we either estimate glomerular filtration rate or we have some sort of structural damage. So I want to jump to metals and CKD. So this is, one of the first things that I would do is trying to understand how metals impact individuals, is to look at some recent population data. So this was a study published 2018 in Current Environmental Health Reports. This study was a meta-analysis looking at publications in PubMed between January of 2013 and April of 2018, looking at those publications that were associated with kidney disease, arsenic, cadmium, or lead. And the initial PubMed database identified 27 publications that the authors addressed renal dysfunction and arsenic, 41 for renal dysfunction and cadmium, and 232 renal dysfunction and lead. And then as the abstracts were reviewed and evaluated for certain criteria that would indicate some level of rigor, and then the full text articles reviewed, there was a systematic triage of publications down to a level where the authors felt that the data could be compared. And so in looking at these data, some of the conclusions, just broad-based conclusions, were lead does have an effect on the kidney and renal function. Arsenic has an effect in some cases, negative effects. These are all negative effects. And cadmium, they couldn't really establish that cadmium had effects for renal dysfunction in humans, but this is my plug for CIEHS and wanting to have people that come through CIEHS and IMFC work with the statistics score. Part of the evaluation of the data were that many of these studies were excluded because of a lack of statistical testing for association of metal levels with kidney damage. So there are ways to statistically evaluate the data to see if there's an association between two observations, or if they're completely unassociated and you're just making a preferred conclusion. So most of these studies were underpowered, meaning that they had less than 100 individuals involved in the study. Very few had, only these few had more than 150 individuals involved in each particular study. And so in human translational type studies, people are outbred. There's a lot of genetic variability how people respond to an insult, lifestyle, environmental modifications, nutrition, age. And so there needs to be appropriate adjustments for those variables. And so in considering those covariate data, you need a large N, a lot of individuals involved in this study. So that's something that CIEHS would be able to help with. And then most of the studies actually did not adjust for covariate data that would be independent features modifying renal function. So individuals with hypertension, dyslipidemia, or diabetes, these are known causal factors for chronic kidney disease. So all of those studies that identified enhanced levels of cadmium or arsenic or lead should have been adjusted for these variables, but weren't. So what that says is that there's a little bit of tough sledding to justify this type of research. But I do want to invoke a fellow faculty member here, a friend, Matt Cave, who was featured on in the cover of the October 2021 edition of NIH News and Health, where he is addressing environmental factors on non-alcoholic fatty liver disease. And so this is a pull out of there that I like. This quote up here is that Matt says, "We're finding that these things probably serve as double whammies." And so essentially all that covariate data could be a double whammy. And so we may be losing a lot of really solid metal toxicologic data on the kidney, but we're losing it because we're not adjusting for covariate data. Also, around the globe, environmental problems are more endemic to some areas than others. And so if you're doing all of your studies in Louisville and the East End, you may not find toxicity associated with renal dysfunction. But if you go to the tropics, Asia, Central Africa, you might find in high levels of problems. So in reviewing environmental exposures to metals and how they impact the kidney, this is a recent publication in Nature Reviews Nephrology by some individuals that really focused on particulate matter, PM 2.5, but they did bring in a little bit of data on pesticides and a little bit of data on metals. And this is a very nice summation, I think, and I'll try to draw your attention to a few things here. One is a lot of these metals are coming from some source of contamination. So we have human activity that are driving these exposures. A large number of these metals are damaging the tubular epithelium, either directly or in the interstitium. And the mechanisms for these damages are oxidative stress, oxidative stress, oxidative stress, or DNA damage. DNA damage in a couple of places, DNA damage. So we kind of know what we're going to be looking for. And so hopefully with a proteomic approach, we can try to drill into this and garner some information that is valuable and useful. There were a couple of authors, Robles Osorio in Mexico, who through the mid-2010s published a series of manuscripts and reviews on lead, cadmium, and arsenic and the effect on the kidney. And most of these were reviews, and I'm not saying that these are the best manuscripts to reference for this data. Dr. Kai and Dr. Young have some really nice ones on these, but I like the flow diagrams, and so that's why I'm using these for the flow diagram. So there's a lot of mechanistic commonalities to the study of metal-induced kidney damage, and you'll see that consistent with the previous table is the exposure of the metal is going to lead to oxidative stress, increase in free radicals, and then activation of transcription factors that are going to drive inflammation and apoptosis. And at other levels, they may affect other second messengers, such as cyclo-GMP or calcium, et cetera. Arsenic exposure driving some caspases, driving some free radical generation, proximal tubular injury. So in a way, we kind of already know through a hypothesis-based approach what to look for. But with proteomics, proteomics is a hypothesis-generating approach. And so those data on looking for free radical damage is a tough sled for us with proteomics because that's a post-translational modification, fairly infrequent. Looking for DNA damage is going to be difficult for us because John Wise has a great method with his DNA comet tail assay. But chemokines, cytokines, typically we're not going to be seeing that. I presented a couple of years ago, 2 years ago, to this group about cadmium, and we looked at it as a phosphoproteomic approach. And with that, we did specifically see signaling pro-inflammatory pathways at the level of protein kinases. This is more of a work in progress for arsenic. And then, although, as Dr. Chin mentioned, I am interested in antimony, but we're not at that level yet. So in this study that I have the kidneys from, these there is some published data on the histology and the animal in these studies by Dr. Kai and Dr. Friedman's group. And so just to run you through the study, the animals, C57 black 6 mice were divided into 2 groups in this study, a control group and a group that had 100 parts per billion, I think it was sodium arsenite, is that correct? In the drinking water. And this was free drinking animals. And so those animals were bred and then the dams were kept on these conditions, and their birth litters were maintained on those same drinking conditions, and they were further divided into groups of male and female, and then those divided into groups that had high fat or low fat diets. And so this study was geared to look at generational effects for the exposure to arsenic, and then on top of that, the double whammy of the effect of high fat diet on the males versus the females. And so this is a publication from Dr. Friedman's group and Dr. Kai's group that examines the phenotypic data on the animals in that study. So this was to look at the effects of arsenic and low-fat diet and high-fat diet on males and females. And what you can see is that, as expected, the high-fat diet, the high-fat diet had an effect. It made the animals fatter. I don't think that was the purpose of the study. That was predictable. But looking at the ratio of the kidney weight to the body weight, what we can see, and based upon the statistics, there was no difference there. So hopefully what we're going to identify from proteomic studies in these animals is going to be a direct effect on protein expression. So pulling more from this data, which was an evaluation of arsenic exposure on mouse kidneys. So this is H&E stain that is staining the kidneys, and within each one of these fields of view, you can see in the middle of it is the glomerular, the glomerulus, a cross-section of the glomerular microvascular bed. And this stain has the ability to identify regions of the mesangium or the mesangial matrix. And so the amount of stain in this region can be an estimation of the amount of mesangial matrix, so the amount of fibrosis. And the blue is nuclei. The rest of this is a tubular interstitial compartment. This is a serious red stain, which is a stain that should highlight collagen as a colored area, a darker purplish blue colored area. And so in these data, it was determined that in males, that high-fat diet had an increase in the mesangial matrix area, and low-fat diet with arsenic as well as high-fat diet with arsenic had effects on the mesangium, the amount of mesangium in both males and females. And the glomerular area itself, there were some effects at the 24-week exposure for males on high-fat diet with arsenic and females with high-fat diet. I think you can see from the purplish color that the arsenic increased the amount of extracellular matrix, the collagen, in the high-fat diet exposed animals, and at 24 weeks there was an exacerbation of that. So again, continuing on with kind of a hypothesis-driven approach is evaluation of the expression of fibronectin. And you'll notice that the high-fat diets, so obesity driving high blood pressure, driving pro-fibrotic environment in the glomerular microvascular bed, you have increased levels of fibrosis, notably in the glomerular area. So this is important because for me with proteomics, because taking a section, 5 millimeter by 5 millimeter cubed area of the kidney, the 90% of the tissue, 95% of the tissue is likely the tubular interstitial area and not the glomerular microvascular area. So with proteomics, we tend to bias our data towards that. So other cytokines and chemokines were ascertained by qRT-PCR, and then hydroxyneonal, looking at oxidative stress, followed that up. And so we took corresponding tissue from the cortex of these animals from the 24-week arsenic exposure, and we processed them for proteomic experiments. And so probably for the first time ever, I'm not going to go through a proteomics experiment, so have mercy on people. Our primary data analyses were achieved using a tool that was designed by Sudhir Srivastava out of CIEHS Rice Group when he was CIEHS Rice graduate student. And so our data analysis that I'll present came from that tool. Secondary analyses were using a tool called Metabo Analysts. And so this is data that Zhenmin Pan generated, where what she focused on was the difference between the male and the female proteome. And so what you can see up here is an articulation of the number of proteins that were identified within this study, the number of differentially expressed proteins, the number of proteins that had an expression a log 2 less than 1. So at a very small expressional difference, but was still statistically significant. And then things that had a larger expressional difference. And what you can see is that we have about 129 proteins that were upregulated and about 114 that were downregulated. So this is a heat map. And if you dug into the heat map, you would see that the majority of these color differences, so red is high abundance, blue is low abundance. So we're driving comparative differences in the proteome based upon males versus females. When we look at this through a multi-dimensional scaling plot, which is kind of a principal components analysis, you'll note that the blues, which are male proteome samples, and the red, which are female proteome samples, are separating across this. So our primary level of separation is based upon just sex itself. When we begin to adjust for sex and look at the effects of diet and arsenic, then the numbers of differentially abundant proteins drops dramatically. So the number of upregulated proteins, 20, the number of downregulated proteins, 11. And when we adjust for multiple comparisons, it's 2 and 0. So very small differences exist within the proteome as a result for the exposure of arsenic. And that's with all the caveats that I've explained before. And so when we begin to look at the heat map to see if there are gross differences in these samples that can, even though we don't have significant differences, are there systematic group differences that define the samples? And we can begin to see maybe there's a difference in the entire proteome, if you will. But when we look at this for a principal components analysis, there's no systematic difference that's allowing the data to self-sort. So that was give me a second. One of these. Oh, sorry, that's male mice. That's female mice. So we speciated out into 2 sexes. So similar to the females, the males have small differences. So Metabo Analysts is software that was originally designed to analyze metabolomics software. It's a group out of Canada, and they've developed ways to have multi-way ANOVA comparisons and some timed repeated measure comparisons, and a little bit some higher level theory than what I'm normally able to do. And so the data were imported in that, and this is a principal components analysis, a two-dimensional and a three-dimensional, to say that we're recapitulating the analysis that we did, the primary analysis. When we add in the third dimension for this principal components analysis, we don't have the ability to further resolve out these samples. So even considering three dimensions of variance in the data, we're not separating out males, females, or with arsenic and diet. So another way to analyze the data. So with proteomic data, I haven't said this, I ignored saying this, but as we have better methods for identifying analytes in our samples, we develop longer lists of proteins. And so then the question of so what becomes even bigger. So what? I have 42,000 differentially abundant proteins. Which one is the most important? So how do I rank order these observations? How do I constrain the data? So one way is to, in an unbiased fashion, allow the data to sort itself into groups of proteins that are providing the most rank order difference in the data set. And so hierarchical clustering is one way to do that. If you can't achieve what you need with this type of approach, the unbiased approach, then you go into a pathways analysis, which is constraining the observations to a given pathway. So when we look at kind of a hierarchical clustering, that is using the data from ANOVA analyses to filter the data. So the top bar for this outlay defines the sex. Green is female, blue is male. The second bar of alternating colors of kind of a tan and a cyan are for high fat and low fat. So female, high fat, low fat, male, high fat, low fat. And lastly, the bars beneath that are for arsenic exposure, no exposure, with exposure. And what you can begin to see is that we do have grouped areas of differences that correspond largely to sex. We do have minor group areas that correspond to diet and diet versus sex, but we don't have clear ones that identify arsenic effects. So we're still trying to ascertain, can we see large effects of the arsenic on the kidney? Constraining the data down to just the top 25 based upon ANOVA value doesn't bring any greater clarity. There is an approach for filtering the data, not by ANOVA, but by interquartile ranges, looking at the greatest differences for the variance in the top 75% of expressors versus the lower 25%. And what we see here is that, again, we're driving mainly all of our differences by sex, and some down here possibly by the combined effects of diet and treatment. So that's to say that the proteome is really not revealing itself. So there is not a lot of great clarity to this data for me to elaborate on what arsenic does to the kidney. So this is a little bit of a work in progress. So now I'm switching to the mode of trying to develop more of a hypothesis-driven approach. So one of the things that we can do is we can look at correlation analysis. So we can take some of those strongest changes that we observed by ANOVA and then look and see, evaluate what proteins were most positively and most negatively correlated with this, so that we can begin to evaluate more of a pathways-driven approach for developing our hypothesis. So here are top 25 peaks that correlate with sex. You'll notice that most of these are metabolic enzymes. A lot of them are associated with mitochondrial function. And so large effects we're seeing in our proteomic data set, in the sex-based data set, are probably being derived with mitochondrial function. When we look at diet, it becomes a little bit less clear. There are some mitochondrial proteins involved in this but there's a lot of smaller metabolic enzymes and signaling enzymes. And lastly, arsenic-based effects, again, a mixture of effects. Jumping into a gene ontology-based analysis, looking at just the sex-dependent effects where we had the largest list of proteins. So when you want to do a pathways analysis, to identify a significantly affected pathway, you don't need to have exhaustive coverage of that pathway, right? What you need to do is to, in a comparative level, look at the data that you're uploading compared to the size of the database you're analyzing against, and then look at what fraction of your uploaded data matches to each given pathway. That gives you sufficient information to do like a chi-square test and look to see if there's a statistically significant enrichment of your data within that you've uploaded. And so, again, sex-based differences, we're pulling out data associated with mitochondrial membrane organization, some of the proteins that were involved in that pathway. So perhaps with the arsenic-based toxicity at the level of the kidney, it might be more appropriate to study specifically the mitochondria, which would impact the study design on how the kidneys are collected and retained or maintained for something like electron microscopy. Cellular compartment differences within the sex comparison seems to be bringing up some RNA binding interactions, which it's known that arsenic has effects on proteins that are involved in RNA binding. And lastly, for molecular function, we have RNA binding again. So I think the data are there that are consistent with animal level data originating from carcinogenesis, mutagenesis, teratogenesis. It's just muddied by the rest of the proteome. So our interim conclusions is that most of the data for the kidney are dominated by sexual dimorphism. Comprehensively, we seem to be affecting multiple ontologic components, RNA binding, mitochondria, energy metabolism. I think what we really need is, as demonstrated by Dr. Kai's group, Dr. Friedman's group, where we're seeing histologically effects at the level of the glomeruli, we need to have better specificity in what we're doing to do these comparisons. And a second thing that we need to do better is to understand what database is used to analyze the proteomic data set. So right now on a different project, we are using laser capture microdissection to isolate glomeruli from human biopsies and study a specific component of human kidney disease, where we take that isolated glomeruli. So here's a microscope. It's both enabled with optics, objectives for visual, as well as fluorescent work. And we can put a tissue section 10 micron, 5 micron, 3 micron thick on the slide, put the slide up on the stage, and we can use a laser to, we can delineate a region that we want to cut out, and the laser will basically ablate that area and drop that tissue down into a receptacle. And we can use that receptacle for whatever types of experiments you want, proteomics, transcriptomics, whatever. For us, it was proteomics, so we feed it through our mass spectrometer, and we look at our proteomic data set. And so this is something that we're considering with the arsenic-based animal data, is to drill down into glomerular proteomics. The other thing that's good about this platform is it does fluorescence-based imaging, and we can delineate those areas based upon fluorescence. And so this is an example of us staining a kidney section with a lectin that allows us to highlight proximal tubular areas, so we can focus on proximal tubular compartment. So spatial resolution is a big thing these days, and you all know about single cell, whatever your favorite flavor of single cell. But for us, using biopsy material, being able to retrieve it by single cell omics is not a practical approach, coming from an formalin fixed paraffin embedded tissue sample. But what we can do is we can stain that tissue, and we can use a stain that actually will allow metals to be highlighted. So this was an example coming from a publication in 2009 where they were looking at quantitative imaging of metals in tissue, and they were looking at copper. So these individuals had an animal model of Wilson's disease where the liver had ATP7B knockout. So this is an ATPase that drives copper transport. And there was a 6-week-old animal that was pre-symptomatic. And so they stained this with this rhodonine stain, which highlights copper. And then they also took that tissue and analyzed it by atomic absorption spectroscopy. And in this image, you see there's very little, according to the authors, very little copper staining. However, the amount of copper quantified by atomic absorption was 277 micrograms copper per gram of tissue. They looked at a 20-week-old animal where the animal was symptomatic for Wilson's disease, and the rhodonine stain did highlight copper deposition, but the amount of copper in the tissue was actually less. And so trying to use a stain like this for driving laser capture microdissection probably isn't the best thing, but there are fluorescent stains. There are many nice publications where people have taken cell culture or tissue sections and stained for using a fluorometric stain that binds to zinc, and you can localize regions of differential abundance in zinc. This is just a human biopsy carried through the same type of a late stage Wilson's disease individual, low levels of copper, but high levels of staining. So sometimes stains are not always accurate. It depends upon and requires the stain to be able to bind and chelate. So there are other ways for spectral, spatial localization of metals, and one way is through X-ray based detection. So I have a KY Embray Award from their EM proposal series last year that I'm working with Michelle Barati and Julia Abersol and Mike Martin at the Micro Nanotechnology Center. And the goal of this is to take kidney sections from the animals that went through the cadmium and the arsenic study and to evaluate they have a electron microprobe that has a detector for fluorescence detection, stimulated fluorescence emission. It's kind of an epifluorescence type of setup, but the X-rays that are emitted are specific for the metal. So we're evaluating whether or not we can develop a spatial localization of metals within animal tissues so that we can then overlay that with histology. And if we can do that, then that'll become part of IMFC's offerings. And the second approach where we need improvement is with proteogenomic methods. So you look at Mayuk or Anna out of Dr. Stace's lab, their data on arsenic and splice variants and carcinogenesis, and it's completely compelling data, right? So we know that arsenic drives transcriptional effects that impact translation. So when we conduct a proteomics experiment, we're relying upon a few peptides to identify the presence of a protein. Whether or not we identify a region of the protein that corresponds to exonyl junctions, splicing junctions, is pure serendipity. And it's serendipitous on multiple levels. One of the biggest delimiters for that serendipity is the database that we use to search our spectral data. The databases that we use are for canonical genes, not for splice variants. But there are processes available for basically taking a sample and collecting proteomic data on that sample and collecting transcriptomic data on that same sample. The transcriptomic data containing those splice variants information, and your database used for your proteomic analysis comes from this. So this is out of Pratik Jagdap's lab in Minneapolis. And so this platform is called Galaxy P. He has one for meta proteomics as well. So this platform allows us to collect data here locally, proteomic data, transcriptomic data, short code, long code, whatever. It was originally developed for short code data. Then that is pushed through a summarization process that allows us to identify confidently peptides from this database. We remove false discovery rate based upon target decoy databases. We end up with a putative list of novel peptides that we can then begin to filter and map onto the genome. And what that ends up with is a listing of peptides that map back to the genome that either give us transcriptional frame variants, so when we have a frame shift, or we have differences in splice variants. So I didn't address the figure that's coming out of Anna and Mayuk's paper that's been resubmitted and hopefully will be accepted in the next imminent future. And Chris talked about this in his item FC presentation the other day about the effect of arsenic to modify how exons are spliced together such that you have skipped exons, mutually exclusive exons. So you have products where you have different exonal patternings, retained introns, and alternative splice sites. So all of these figures, barcoding figures for a proteomics person would represent data in one experiment where we might identify a peptide, in other experiments we don't identify that peptide. So that might be considered a missed peptide in that experiment, but the peptide may not be there because it may be spliced out of the protein. So we have two approaches for improving the sensitivity and specificity of what we're doing for arsenic-based research that I hope that within the next year we can establish. So a variety of people. The original people in this study, people in proteomics and microscopy cores. The initial work was performed by Josh Carty as a DIR medical student. He's now at Vanderbilt. I was hoping he would stay here, but he's a good guy. He's going to have a good future. Informatics, Shesharai and Janmin Pan. The award from KY Embray and MNTC to do the epifluorescent work, and then the funding sources going back from the first start of all this. So I'll be happy to take questions in the next 3 minutes.
So you talked about the requirement of adjusting for covariates like hypertension or to understand the effect of the method. But given that, for example, arsenic increases the risk of developing all of these diseases, they're not really independent covariates, and as such, can we really adjust for them?
So Mayuk asked the question of the need to adjust for covariate data, and that arsenic affects all of these parameters. So do we really need to adjust for covariate data? And so the covariate data I'm talking about is not So our endpoint for our analysis of does arsenic have an effect on an organ is an endpoint of organ damage. And so arsenic, I don't think affects serum creatinine levels and ultrafiltration of serum creatinine.
Actually, it does.
How?
I don't know, but it does. Like there are multiple publications, which is why there is a big discussion going on whether people should use creatinine and correcting arsenic damage as the tool.
Okay, so good question. The covariate data that you were talking about, though, was adjusting for hypertension.
Which again, arsenic would.
Dyslipidemia, diabetes.
Yes. Oh.
So, yeah, you've got me stumped on this one because my initial answer would be the order of magnitude of the effect of arsenic on those. That order of effect is different than the order of effect of the disease on the renal function. So I think it may be a distinction without a difference or true unrelated. So arsenic does have an effect on diabetes, but that doesn't say that everybody that is exposed to arsenic develops diabetes.
Definitely
right? So the primary effect of arsenic is not to induce diabetes, but diabetes is known across the board to have renal effects. Hypertension is known to have renal effects. So, I mean, like 40% of blood pressure control is through the kidney. The number 1 or 2 drugs for treating high blood pressure actually the targets are the kidney. So there is a direct corollary for that. So I'm not going to answer your question, but I did answer your question.
I think it's Dr. Hine raised the hand.
Okay.
You can talk. Hi, Mike. This is Dave Hine. Can you hear me?
Yes, sir.
I enjoyed your talk. It was a great talk. About 10 slides back, you had a slide where you showed hits for metabolic enzymes. Can you find that slide? Yeah, that's the one. That's the one. Correlated with sex, there's a particular enzyme near the bottom that's cut off,
Yep.
and that I have a particular interest in, and I'm wondering which N-acetyltransferase that is.
I can email you.
Okay.
I don't know which one that is.
All right. I'd be very interested to know. And then you've got acetyl coenzyme A up there as well, which is not an enzyme. So what are you?
So this was cut. Sorry, the character space was truncated.
Okay, so it's an enzyme there.
Yeah, the full name for some of these is truncated.
Okay. All right. Well, I look forward to getting your email. It was a fascinating talk, and depending on what you tell me, maybe we'll have some interesting discussions.
Absolutely. I'll copy paste the list into an email, and you can just look at all those.
All right. Thanks.
This may speak to the naivety of my background in proteomics. Sorry, I said that. Idea, you talk about canonical proteins and the idea that that's the output. But what about misfolded proteins? Like how are those able to be detected with the proteomics approach?
Right. No.
No.
So I mean, what you're discussing is referred to as a proteoform. That an isoform or proteoform. So let's say you use the word globin to describe a protein, or you say heat shock protein, or you say some protein of which there are multiple isoforms. They may have a common active domain where the amino acid sequence is identical. So let's say that the only peptide out of that protein was identified from that domain. So the algorithms that we have will assign an archetypical gene as the parent for that family. So unless we identify a peptide that is proteospecific, we won't identify the variant. Now, as far as a proteoform, there are people that use effectively a footprinting method to try to unfold a protein, digest it with a limited digestion approach, identify peptides that are liberated, and begin to deduce what's been the domains that are active for proteolytic digestion.
But you have to go with a specific protein of interest or isoform.
Yeah, you would.
Can't do like a global approach to see
Uh,
a group of misfolded proteins.
Yeah, I mean, there are 1 or 2 labs that are doing that. It's not a routine level of analysis. It's very empirical, very experimental, where people try to map out what's accessible for proteolytic digestion. Yeah. So Well, thank you for your time.